So this is the final exam review for Biology 1010, and I'm going to go through each and every lecture. And for the most part, it's pretty much one lecture per question on the final exam. The exceptions are lectures 1 and 17, and that's because those are kind of the bookends. Lecture 1 is the overview of everything we were going to learn, and lecture 17 is wrapping things up from everything that we've learned. And so those are kind of the two main ones that I gave some extra questions on. Now, it doesn't mean that there's only one concept per lecture you're going to be tested on. In fact, each lecture has many concepts that I want you to take home with you from this course. But for as far as how many questions you'll get, they're grouped together so that you're only going to get one of those. For example, here in lecture 1, as we begin, you're going to have two questions on these 5 concepts. And when we get to other lectures like lecture 2, you'll have one question on any of these concepts, and lecture 3, you'll have one question on any of these concepts. And so there are multiple questions that are possible, but as far as the review, you'll only get one on the exam per lecture. And the exception is this first lecture as well as lecture 17. So let's begin. So as we learned back in lecture 1, we learned how living things are organized, use energy, maintain homeostasis, reproduce, and evolve. And so they're going to be very similar to what you studied and learned for quiz 1, but they're not going to be exactly the same as what was on quiz 1. So it's not one of those things where you have to go back to quiz 1 and kind of look up all your notes. And the reason for that is because with a lot of these final exam questions, I'm going to pull from concepts that we've learned throughout the semester to bring it home. Meaning, in the beginning of the semester, when we talked about evolution, we really only talked about natural selection because that was the most common thing that people knew. But now you know the Hardy-Weinberg equilibrium and the 5 principles of evolution. And so if I mention the Hardy-Weinberg equilibrium and how there are 5 different principles that are part of this characteristic of living organisms, then you know that's evolution. So they're not that much more difficult, but they are going to incorporate things and concepts that we have learned from the other lecture. For example, reproduction. Now we've talked about mitosis and meiosis, so I may just simply say mitosis and meiosis are examples of how living things, and then you'll choose reproduce. Or energy use, if I talk about photosynthesis and glucose metabolism, those are lectures 8 and 9. You're going to find that those that I may just say those concepts, and you're like, oh, that's how living things use energy. Or I may say cell theory is how living things are what, and you would say organized, because that's basically the level of organization is all living things are made of cells. So homeostasis is kind of like what we learned before, where I may give you an example of thermal regulation. You get too hot, you sweat, you get too cold, you shiver. It'll be simple examples of how we regulate our bodies or how living things regulate to stay alive. So that's one question. It could be any one of these 5 concepts. The second question is more of a fill in the blank. It's still a multiple choice question, but the answers are going to be these concepts like cell theory, photosynthesis, glucose metabolism, mitosis, meiosis, Hardy-Weinberg equilibrium, and I'm going to describe that concept. I'm going to describe and define cell theory, or I'll describe photosynthesis, or I'll describe glucose metabolism, or I describe sexual reproduction, or the principles of evolution, and I'll say Hardy-Weinberg equilibrium and things like that. So it's very similar to this, but it will have some of the main concepts that we discussed regarding these 5. So that's really the two sides of the same coin. And those will be the first 2 questions. So on your final, they will be in the order that we learned them, meaning questions 1 and 2 will come straight out of lecture 1, and then question 3 will be the next one, question 4 will be the next one, and 5, and so on and so forth. Okay, so it makes it really easy for you to kind of follow along, so you don't have to jump around and you're not having things here and there, and it just keeps things simple for you to be able to take the final exam. Okay, lecture 2, which was on the scientific method. The main thing I want you to leave this class with is an understanding of the difference between a theory, a principle, and a law. And now we've talked about many of these theories: cell theory, theory of evolution. We've talked about the principles of evolution, such as natural selection, mutation, sexual selection, genetic drift, and all of that. And we've talked about a couple of laws, law of inheritance, law of segregation, law of independent assortment. And so basically I'm going to describe a theory of evolution. I'm going to describe maybe the principles of evolution, such as natural selection and mutation, or I'll describe the laws of inheritance, and you'll have to choose, oh, that's a law, oh, that's a theory, oh, that's a principle. So these main concepts that we studied throughout the semester will typically be those concepts. If I say Hardy-Weinberg, you know that that's the principles of evolution. So anything that has to do with Hardy-Weinberg equilibrium, those are the 5 principles of evolution. If I explain why there's so many different species on our planet, that's basically the theory of evolution. It basically explains why there's so much diversity of life on our planet. And then, of course, we talk about the rules that regulate inheritance of genetics and alleles, and we talked about that in lecture 15. Again, those are the laws that govern inheritance in biology. And so those are the 3 concepts that I'll test you on for your final, and you just got to know the difference between those. As far as lecture 3 with atoms, the main take-home is one of these 3 types of bonds: covalent, ionic, and hydrogen. I would pay more particular attention to covalent and hydrogen because these 2 bonds were the most important that we talked about in biology. Remember, covalent bonds is the type of energy that keeps all of your molecules together, and that's typically what we call potential energy. Glucose and fats and proteins and all the energy that exists between the atoms that's stored, that's covalent bond energy. But we can't discount hydrogen bonds, because remember, these are the weak bonds that give DNA its properties and proteins their shape, water its properties, and things like that. So we can't say, well, because it's weak, it's not important. In fact, it's pretty important because of all of the different characteristics that molecules take on due to their polarity and the hydrogen bonding that exists, especially when it comes to some of these biological molecules like DNA and proteins. So I will describe the role that one of these bonds plays in biology, just like I just did right now, and you will tell me what type of bond that is. And so that's what I'm going to test you for the final exam. Lecture 4 is again going to be basically solvent through density. I don't have any questions on acids and bases, acids and alkalinity, nor on metabolism. I just keep it to these first 6 right here. So this one is one of those where you definitely just want to go back to your notes in lecture 4 and kind of review over the examples that we talked about. This is one of those that's almost exactly like quiz 4. So you're going to want to refresh your mind on those notes from those 4, or from lecture 4 for these 6 concepts right here. Lecture 5, these are going to be your answers: carbohydrate, lipids, proteins, nucleic acids. And essentially I'm going to describe some of the molecules' importance for biology, and you're going to tell me which group that belongs to. So let me just kind of refresh your mind on some of these groups. Remember, nucleic acids, though, that's the genetics, that's the inheritance, that's the information that all living organisms use to make all of their proteins. This is the DNA and the RNA, and so make sure you understand that. Proteins, again, this is what does all of the work. These are the things that actually do all of the work in the cells. They move things across the membrane, they metabolize things, there's enzymes, there's transport proteins. I mean, this is ultimately what does everything in biology. Everything else is either information, structure, or energy, but proteins, these are the ones that actually do stuff. Lipids, remember that lipids play an important role in energy and structure. I'm going to focus primarily on structure, especially phospholipids, because if you remember, we talked about how all cells have that phospholipid membrane, and that belongs to this group, and that's one of the most important biological molecules for cell biology. And so that's what I'm going to focus on for this group. Carbohydrates, remember, this is energy and structure. Things like glucose, but also things like cellulose, structural carbohydrates that make up things like fiber. And so I'm going to focus on some of the less well-known things. I'm not going to test you on any of the different characteristics of these four groups, but I'm basically going to give you an example of these four groups, and you'll have to choose which one it belongs to. As far as the organelles go, this is lecture 6 when we talked about the cell biology. I'm going to have the organelles as the answer, and I'm going to pull from a lot of the concepts that we learned throughout the semester. And so let me just kind of give you a shorthand. You can go back and kind of refresh your mind on a lot of these, but some of the main concepts that we learned throughout each lecture has to do with each one of these organelles. So I'm going to kind of give you some key words as we go through here so you know what belongs with what. For example, the nucleus, that's where all the genetic information is held. In lecture 10, we talked about the process of copying the DNA, and this was where transcription occurred. And remember, there's two forms of transcription, but it doesn't matter which one you use. Transcription, all transcription occurs here in the nucleus. So if I use the word transcription, you know I'm talking about the nucleus. The ribosome, this is where you'll hear the word translation, right? This is where the ribosome would interpret the RNA information and make the protein. And so it would literally be translating from one language to another. And that was also a major concept of lecture 10 that we learned. So ribosome translation. Don't really have any questions on the rough ER and the smooth ER. Golgi apparatus, remember, is that modification organelle. We did talk quite a bit about antigens, which are the surface glycoproteins, right? When we talked about blood type, now we talked about blood type A and B and O. That's where the Golgi apparatus comes into play. That's ultimately the one that modifies the proteins and attaches that oligosaccharide onto it, thus giving you the surface proteins with the carbohydrate, which we call glycoproteins. So that's Golgi apparatus, especially when it comes to cellular recognition. Lysosome, this was highly associated with cancer, especially when we talked about cells undergoing apoptosis and how apoptosis and lysosomes go hand in hand, especially when the cell is digesting itself from the inside out when it goes through that process. I don't really have any questions on central vacuole peroxisomes. But definitely chloroplast and mitochondria. Why? Because chloroplast was lecture 8 and mitochondria was lecture 9. So you better pay close attention to those because they were important concepts. Remember, chloroplast has to do with autotrophs, photosynthesizers, anything that gathers sunlight and turns it into energy, potential energy like sugars and things like that. That's chloroplast. Mitochondria, remember, this is processing food. This is processing covalent bond energy into ATP. And this is also in plants. Plants don't just have chloroplast, they also have mitochondria. So mitochondria has to do with breaking the covalent bonds down and turning them into more usable form like ATP. Cytoskeleton, you'll see this if I talk about mitosis or meiosis, especially when it comes to the mitotic spindle, which forms both during mitosis and meiosis in separating and sorting and organizing the DNA. Those are lectures 12 and 14, so you can bet that I'm going to bring that up. And then, of course, cell wall. We talked extensively through the semester about how cellulose, chitin, peptidoglycan, these are all different types of cell walls that cells use to protect themselves. And so any one of those that I just mentioned are typically answers on the final exam, and I'm going to describe their role just like I just did, and you'll have to tell me which organelle that corresponds with for that process, be it photosynthesis or metabolism or blood type like the Golgi apparatus or transcription in the nucleus. Okay. Lecture 7, there's a lot to that, but primarily I'm going to have you take home membrane transport. Now, this is one that's very much like what you already learned in Lecture 7. So go back to your notes, review over simple diffusion, facilitated diffusion, osmosis, review active transport, exo and endocytosis. Any of those are possible on the final exam. They're very similar to what you've already had in Lecture 7. So go back to your notes and review over all of those. Lecture 8 is primarily on this equation. I'm not going to test you on any of the smaller particulars of the Kelvin cycle or some of the differences between photosystem 2 and 1, but there are a couple things that I need to make mention. So make sure you understand, again, what all of this means. Basically, carbon dioxide combined with water and light is what's used to make sugar or glucose and oxygen. Now, if I ask you again what water is used for, that's one of the possible questions. Remember that water doesn't go into making the sugar that much. Yes, the hydrogens do, but that's not the bulk of what makes up the organic mass of the plant. See the carbon dioxide right here, the CO? That's what makes up most of the mass of the plant. So again, the role of water is essentially the splitting of water and turning it into oxygen gas and stealing the electrons. Those electrons go into making the covalent bonds here in the sugar. So make sure you understand two major factors. First off, that the organic mass of the plant primarily comes from the air, and that water's main role in photosynthesis is the electrons are used to actually covalently bond all of these atoms together. Most of the mass of water, like this oxygen right here, that actually goes into the air, and that's what you and I breathe in and what we need. So those are the two major concepts that I want you to take home from this for your final exam. Here, I mainly want you to understand the difference between these three forms of metabolism. So this is lecture 9. I'm not going to get into the particulars of the Krebs cycle or the electron transport chain. I merely want you to understand the fundamental difference between these three. So remember, aerobic respiration is the most efficient way of getting energy. It goes through the mitochondria and ultimately uses oxygen, which is why it's the most efficient, and that's where you get most of your ATP out of it. Anaerobic is the next efficient, and fermentation is the least efficient. And the reason for that is because you really don't extract any of the energy from pyruvate. Most of it's lost through the fermentation process. You're only getting energy through glycolysis. That's it. So fermentation is definitely the least efficient way of metabolism because you're not even getting any energy out of this process here. Now, lecture 10 is transcription and translation. Basically, this is going to be on these three concepts. That's it. And most of the other stuff that we learned is related to some of the other concepts throughout the semester. But make sure you know the difference between DNA transcription, RNA transcription, and translation. These are generally what you're going to be tested on. I'm going to talk about what purpose they have. So if I say which of the following processes is only used for cellular replication, for mitosis and meiosis, that's DNA transcription. Or if I say which of the following processes is used before you make a protein, that's RNA transcription. Or which process is used to convert the RNA, which is the nucleic acid sequence, to the amino acid sequence, that's basically translation. So those three concepts are the three that you'll be tested on for your final exam. So lecture 11 is biotech, and these are the concepts that I'm going to teach you or test you on. These are going to essentially be the answers. Transgenics, DNA sequencing, DNA fingerprinting, DNA microarray, and stem cell cloning. And so I'm basically going to define them. I'll define what a transgenic organism is, and you'll choose that. I'll describe the difference between DNA sequencing, fingerprinting, or microarray, or talk about cloning. So they're not too difficult. The only part is these three right here. So let me review over the DNA sequencing, fingerprinting, and microarray. Sequencing is spelling it out, basically letter by letter by letter, base by base, so that you can find new mutations. That's the whole benefit of sequencing. Fingerprinting, you're not actually analyzing the coding part of your DNA. You're just looking at parts of the DNA that give very specific segments that can be used as an identifier, but you can't tell whether somebody is going to get cancer or have a predisposition to some other problem. And so you really don't know any health information, but it is useful for things like paternity tests and for forensics and disaster identification and things of that sort. Microarray, remember, these are the cheapest way to analyze the DNA. They're used for Ancestry and 23andMe and a lot of these companies. You have to know what you're looking for. And basically, these are SNPs, or single nucleotide polymorphisms. Basically, the most common mutations in the human genome, and they're able to look at millions of these very simply, very cheaply, which is why we still use them today. The downfall is you can't really discover new mutations because that's not how these chips are designed. They're generally designed to look in very specific locations for the most common mutations and the most common health problems that people have. They can be used for things like Ancestry, but there's a lot of health data in there as well for therapeutics, for cancer treatment, as well as possible cancer therapies and what causes the cancer, but they have their limitations as well. So that's the microarray. And of course, stem cell, these are the embryonic and adult stem cells that are usually used for therapeutic or reproductive cloning. So it could be any one of these terms, and I just want you to be familiar with all 5 of these, but you only have one question again on your final for this. This one's easy. There's only one question, and it's pretty simple, and it basically asks what's not a purpose for cellular reproduction. Well, you're looking at all 4 of the purposes. So what's not up there? Sexual reproduction, right? This is the only thing that cellular reproduction is not used for. All these others are what you and I use. Some species do this. They clone themselves. But growth, regeneration, repair, renewal, all these things are for mitosis. But it's cloning. That's essentially what cellular reproduction is. Your cells are repairing using your own cells, and the cells that are used to repair are no different than the stem cells that are being used to repair. But when you get into sexual reproduction, that's a way different process. So that's what you'd be looking for in this one. For cancer, with all the things that we talked about, really the 2 main things to focus on are these 2 groups of genes. I figured that this was kind of the most important part to take away from this lecture, and that is what genes are the most prominent ones that regulate the cell cycle and thus lead to cancer. Not all cancers are caused by genetic problems, but many are. And so make sure you know the difference between proto-oncogenes and tumor suppressor genes. Pretty much like we learned back in lecture 13. Make sure you know that the proto-oncogenes promote the cell cycle and prevent apoptosis, while the tumor suppressor genes work in opposition to that. Of all the things that we learned in sexual reproduction, the one I want you to take home with you is non-disjunction. This is what I'm primarily going to focus on. Remember, this is a process that can occur in either anaphase 1 or anaphase 2. It's a failure for either homologous chromosomes or sister chromatids to separate, and that leads to either one more or one less chromosome. And the most common form, which we talked about, was Down syndrome, such as having an extra chromosome 21. There were other chromosomes that we looked at in the sex chromosomes that led to various syndromes as well. But primarily, I want you to know what non-disjunction means, when it can occur, and one of the most common cases of non-disjunction, which is Down syndrome. For lecture 15, no Punnett squares, yay! But you are going to have to know all of these terms. And just like we learned back in lecture 15, I'm going to test you on the same thing, where I'm going to look at describing the relationship between genes and traits and alleles and traits. And these are all possible answers that you're going to use for yours. So make sure you review over all of these. Again, incomplete dominance and codominance are really the only 2 that people tend to mix up. Incomplete dominance is basically when both alleles mix together and give a combined phenotype. We call it an intermediate phenotype. Codominance is when you get both phenotypes simultaneously. And the only example I ever gave of that was blood type A, B, because you're both blood type A and you're blood type B. So 2 phenotypes with codominance. Incomplete dominance, we talked about the flowers. We talked about the pink and being the intermediate phenotype being combined from the red and the white. So make sure you know the difference. All the rest are pretty straightforward and simple. So go back and review over your notes on all of that. Hardy-Weinberg equilibrium. Basically, it's that question that you had on lecture 16 where I asked you what causes evolution. So the Hardy-Weinberg equilibrium no change scenario, make sure you know this because it's just like in lecture 16 where I'm going to say, oh, non-random mating. Oh, there it is. That would cause evolution. Or the population is small. There you go. That would cause genetic drift. Or that there is natural selection. Or that there are mutations. So make sure you know the 5 conditions of the Hardy-Weinberg equilibrium. The question will basically say which of the following will cause evolution to occur, and it will deviate from one of these 5, and you'll choose that one. Now, lecture 17, this was the one that I told you that there were 2 questions on. The reason for that is because it kind of sums and wraps everything up. And if you're unsure of how you're going to be tested on this, you need to go back to the review for lecture 17. At the very end, I went over all of the general characteristics of all the kingdoms and domains of all species on the planet. I wrote a list of the bacteria, archaea, fungi, animals, plants, protists. I talked about how they're single-celled or multicellular and single-celled, whether they reproduce sexually or sexually and asexually, and all of that. So go back to the review on lecture 17 if you missed that. I gave a whole long list of that. This is basically how you're going to be tested on the final exam. There'll be 2 questions on those concepts. So make sure you review over those because I'm going to describe species out of each kingdom or domain, and you tell me which kingdom or domain I'm talking about. Again, the answers will be bacteria, archaea, fungi, animals, plants, or protists, right? This huge group, this huge kingdom right here. And then your final question on your exam is going to look primarily at the ecology, ecosystem. It's going to look at these 4 on the pyramid and this 5th one: producers, decomposers, primary, secondary, and tertiary consumers. So make sure you know the difference between those and the roles that they play. And that one's pretty much the same as what you had on lecture 18, pulling from those questions where it may say a bird eats an insect, which eats a plant. The bird is considered a what? And you would choose secondary consumer. Or I may talk about organisms that break down the dead and the waste and they recycle nutrients back into the soil. Those are decomposers. Or I might say these groups of organisms bring nutrients from the soil. They reabsorb these nutrients. They bring in light and they produce their own food. Obviously, I won't use the word produce, but they're going to make their own food, and these are the producers. So that will be that final question on your exam. Okay, so you only get 1 chance at the final. Unlike other quizzes where you generally have 2 chances, you only have 1 chance. So take your time, review over this material, ask questions, make sure that you are ready for it because you get 1 shot at this, and that is ultimately the grade you're going to get, at which point you'll have summed up everything that you've done for this biology class.